Yeah.
So the last talk is given by Pol del Aguila Pla.
It's fine.
It's going to be about the particular method for cell differential.
Yes, so my name is Pol del Aguila Pla, and I come from KTH, from the Department of Information Science and Engineering, Electrical Engineering, but also from the Access Linnaeus Research Center. So yes, I want to first acknowledge that all the work I will present would not have existed without my supervisor, Professor Joachim Yalden, and the funding he secured from the Enterprise MapTech and the public agency in Sweden, VR. I also want to acknowledge the workshop organizers for the amazing opportunity, and then the KTH Opportunities Fund and the Newton Alice Wallenberg Foundation for travel funding. So let's begin. 2 years ago, an enterprise comes and gives us this problem. They sell these things in which there are 96 wells, and inside each of these 7-millimeter wells, a biochemical experiment happens. And the output of each of these experiments is an image such as this one. And the biologically relevant thing here is how many spots are there in that image and which are their precise locations. These spots are about 200 micrometers. But of course this is an ill-defined problem, because you say, what is exactly a spot? Does it have a precise location? Does it have a size? So what we really have to do is to find active cells. And active cells are defined as cells that generate a certain kind of particles the assay is tuned to detect. Let's have a look at a close-up of what is happening in there. There are certain cells stuck to the bottom of the well that generate some particles. And these particles, they move in a free Brownian motion across the medium. And if they collide with the surface, they may get stuck. And this process is known as adsorption, and it's signified by these little black crosses. And after a while, they may escape and just keep on with their Brownian motion, which is called desorption. And the only particles that will be imaged will be the ones that at the end of the experiment, at a certain time t, are stuck to the bottom of the well. So let's break a bit the simplicity of this drawing and go to a more realistic example. This is an approximated Monte Carlo simulation of just 1 second of one of these assays. These assays tend to last at least 24 hours. And when this video finishes, we're going to have the image if we did only 1 second and we're capable of detecting that much, that few intensity of signal. And yeah, so now it's not interesting anymore. Like this. So it gets already, with this simplified simulation, it gets already the point across. It may be very difficult to find the little yellow crosses from this image data. So let's have a look at the continuous model. If we want to model this physical process continuously, we need 3 quantities. First is density of bound particles at the bottom of the well. Second is it's the density of free particles that are floating around in the medium. And third is the source density rate of new particles being added to the medium by these cells we want to find. It will be spatially sparse in the image domain, of course, because there are few cells compared to pixels. And this will be regulated by this, let's see if I say this right, reaction, diffusion, adsorption, desorption equation. So the first part there is the one that regulates Brownian motion or diffusion. And yeah, the second one regulates or partly regulates this adsorption and desorption. And the third one completes this boundary condition and incorporates this source density rate I was mentioning. This model was known before, and our first contribution here was to be able to write the image observation, so the density of bound particles at time t, as this. So here the G sigma inside is a Gaussian convolution operator in space, and the function A has a very specific structure, which is this one. I'll tell you later how we managed to get this expression. And what's important here is that this A function is an equivalent to the source density rate, that maintains all spatial characteristics. So I wasn't really interested in knowing when the cell had generated the different particles, but I really want to know where the cell is. And the important thing is, whatever this function here is, it doesn't depend on spatial properties, so I will keep those. Because we're already thinking on the inverse problem setting, in which we will want to recover A, we already here start introducing these conditions on that our image has to be non-negative, that we introduce a weighted norm in its space to be able to weight differently errors at different positions. And then we also introduce that this A function will be non-negative, which is coherent with this phi, or phi, that is non-negative too. And it will also have like a masking function for us to be able to precondition the solution to the inverse problem, saying here you cannot place active cells. So this operator A popped out there from this new expression, and because of what it does, we will call it the diffusion operator. How did we derive this? So the first was to think that Brownian motion in the 3 spatial dimensions was independent. Then to realize that the absorption and desorption mechanism only interacted with the movement in the z dimension, and then to see that that meant that the x and y movement would only depend on the total time each particle had to travel freely. So it really doesn't matter if the particle has been traveling, it stops and it releases again. What we care about is the sum of these 2 times that it has been diffusing. And these, based on some previous results that were available on the characterization on the first time to adsorption, we were able to derive this phi function. Finally we changed the variables from this total time of diffusion to something that had more meaning in terms of spatial dimensions, so sigma. So the first application of this, directly, without anything else, is that we can generate synthetic data that looks as similar as this. A disclaimer here is that this image is generated by a fluorophore, and then it's captured by an optically filtered wavelength and a color camera behind it, while this image is just a grayscale image that we generated using our model, loaded into the green channel to make better comparisons. Another disclaimer, this image includes an extra blur to model an imaging system that was not perfect, and also additive noise. So keeping on, of course, the biggest application of this will be to set up this inverse problem. Here we use the weighted norm I mentioned before for the data fidelity term, and for regularization we use a group sparsity regularizer. So this group sparsity regularizer is enhancing or favoring continuity in the sigma domain and sparsity in the spatial domain, and it's actually weighted. This weight is a non-negative function that we can use for many things. So we could, for example, if we remember that A is coming from this source density rate S, we could reweight the sigma space in order for it to be equivalent to a uniform weighting through time in that S. Or more simply, we could, if we put in Xi we put an indicator function behavior, we can choose not to regularize some sigmas, which in imaging it makes a lot of sense, because you could take, for the big sigmas, you could just not regularize, and that would help you predict some background patterns in the image that cannot be explained by cell generating particles. So to be able to solve this problem, we needed to by the accelerated proximal gradient algorithm, we needed to characterize first the diffusion operator. So first in terms of its norm, which was easy because the weighting function is bounded, and then Jensen's inequality and all that gives you the bound easily. And then in terms of its adjoint operator, which also was quite easy to derive from the fact that the Gaussian blur is self-adjoint. So moving on, we needed to derive the non-negatively constrained group sparsity regularizer, its proximal operator. Okay. So here we weren't able to find this in the literature. We know that the group sparsity regularizer was known, but then if you put these two addendums, just the weighting and the non-negativity constraint, we weren't able to find it. But in order to just prove it ourselves, we just followed the same path as the usual derivation of a norm, of the prox of a norm, which is basically we went to the to the differential conjugate, and that led us to the dual norm with an inverted weighting. And then we had to project on the dual ellipsoid, and through Morose decomposition finish the proof. Of course, in a generic case, if you just let the weighting function be anything, that creates a problem because you have a projection on an ellipsoid in there, which you cannot solve in closed form. So for this presentation, at least, we'll keep to the simple case in which our weighting function in sigma is just an indicator function. And then it has this very nice shape in which we regularize just the sigmas that are in a set Aleph. And basically, the sigmas that we don't regularize just get the non-negative projection, while the sigmas that we regularize get first a non-negative projection and then a group sparsity regularization effect. So, yes, to summarize, we have this the accelerated proximal gradient algorithm for functional inverse diffusion. So you may have noticed that I haven't discretized the problem at any point. So at some point we needed to do that, and we decided to do that after having formulated the algorithm, which may not be that traditional. But then the spatial grid we borrowed from the sensor camera, which is imposed on us. And then the sigma grid, we chose it to be non-uniform. So that allowed us to give more detail to the discretization of the sigmas that were smaller, which corresponded to smaller patterns in the image, and less detail into the big sigmas. Then, yeah, we did all this to discretize the operator. We used the inner approximation paradigms as step constant functions. And we chose to normalize the restriction and extension operator so that the norms in the discretized space and the non-discretized space were equivalent, which makes everything much easier. And yeah, so the resulting algorithm of discretizing this whole thing can be argued as the APG algorithm on a discretized model too, if you do it well. So getting back to our initial problem, we have to find those active cells, and a traditional measure to say whether you're finding them well is the F1 score, which is like a compromise between precision and recall. So what we did to generate this plot was to generate 50 images of 255 by 255 with 750 random active cells in there, and then just blur the result by an imperfect camera observation system and add some noise, and then on that image perform 10,000 iterations of our algorithm. Once that was done, since the recovery was not completely perfect, we calculated the contribution of each pixel to the group sparsity regularizer and thresholded that by the value that gave the best F1 score. And we averaged those 50 results for every point in the graph. So I'll explain a bit. So the green and black curves both correspond to our algorithm, but in the discretization, the kernels that output from there are not separable. So what we did was approximating them in the black case by a rank 1 kernel, so separable, and in the green case by a rank 3. So these results pretty much show that it's not worth to go upper than rank 1 unless you really are looking for the fine details. And then the other curves, the blue curve is just applying the convolution procedure to which we give the blur of the camera system, and we also let it run for 10,000 iterations. The red curve is picking maxima on the observed image directly with noise and everything. And the brown dot is just picking maxima in a version of the image without any noise. So yeah, that's our results. And I'd like to take the chance to also thank the people at the mathematics department at KTH that helped me a bit with discretization of operators, Dr. Hollier Core, Dr. Johan Carlsson, Ozan Oktem, and Axel Ring.
So what are the conclusions that are drawn then from the position of the cells? So if you then what does the company do?
Yes, so yeah, they've incorporated this in their workflow and developing a new product now. But the thing is, they compare different experiments in the same set of cells for different quantities, different kinds of particles, and then they want to see how many of the cells there are secreting several substances at the same time. And these proportions they use, for example, researchers to develop vaccines. You want that most of the cells that you throw in there respond to the vaccine by emitting some cytokines, which is a kind of molecule. And then you want them to emit several cytokines at the same time to fight a specific disease. So yeah, that's how it's used. Yes?
Can you say again what you said about this combination of group sparsity with positivity?
Yes. Let's see. Here, yes. So, which part?
I didn't really recall, but there is a result that says that prox have a sum of two functions if one of them is separable. It's the prox of the prox.
So, yeah, this thing is basically that we regularized only the certain sigmas by putting this weighting function as an indicator function of the set Aleph. And then the specific result for this group sparsity regularizer with a non-negative constraint, it says that on the sigmas that the weighting function is not active, you just get the effect of the positivity non-negativity constraint there, while in the other sigmas you get first the non-negativity constraint and then the usual group sparsity regularizer. What do you mean?
There's a result that says if the prox of the sum of two functions can be computed. If one of them is separable, you can compute first the prox of the other one and then apply the prox of the second, of the one that's separable. But for example, if you have some prox and indicator of, say, first order, you can first compute the prox and then just project on the first order.
Okay. In this, I don't know the result enough to be able to check, but in this case it would contradict that, so yeah, I'm not. The papers, 2 papers on this are on arXiv. If someone wants to check. Yeah.
So let's thank the speaker again. This is the